All right. Hello, everyone. Good morning, good afternoon, good evening, wherever you are. It's a great pleasure to welcome you on this new edition of the Worldwide Neurodevelopmental Forum. Our guest today is Claude Desplan from NYU. Claude, hi. Great to have you in. As always, I'll start with a little bit of an introduction of Claude's or of our guest's path and achievements. And then the stage will be fully his for 50 minutes or so, after which each of you will be able to ask any question you're interested in relating to Claude's work. So Claude is trained as a biologist, or originally trained in France, and has been the Silver Professor of NYU's Department of Biology since 1999. So as most of you are aware, his research centers on understanding the developmental and the development and functioning of the visual system, in particular the one that underlies color vision using Drosophila as a model organism. Claude trained, so as I was saying, in France at the École Normale Supérieure, and then received a PhD from the INSERM, did a postdoc in Pat O'Farrell's lab at UCSF, where he worked on homeodomain proteins. He was the first one to demonstrate that the conserved signature of many of these developmental genes is a DNA binding motif. So in 1987, he joined the faculty of Rockefeller, was a Howard Hughes Medical Institute assistant and associate investigator, and then he pursued here, at that time of his career, structural and functional studies of homeodomain and paired box DNA binding domains, in particular looking at the evolution of axis formation in insects. And so he's been at NYU since 1999. His lab has done, as again, I'm sure all of you are aware, has been doing seminal work in the past decade or so on the molecular mechanisms that pattern the fly color sensing photoreceptor neurons. Showed in particular that not everything is deterministic, that combinations of transcriptional factors and stochastic decisions contribute to the diversification of photoreceptors. And perhaps more recently, at least more recently to my knowledge, also provided really interesting insight on the role of lineage in neuronal stem cells, and how spatial and lineage factors interact to define cell types and circuits. As you may also know, Claude not only focuses on flies, but Claude's lab uses a number of interesting evo-devo approaches, including using wasps and ants to study his topic of interest. And of course, together with these remarkable scientific achievements, he's the member of a number of advisory boards on the AAAS, a AAAS member, a foreign member of the EMBO, and has been awarded his most recent award was actually last year, the Edwin Grant Conkling Medal by the Society for Developmental Biology. So Claude, it's really a great pleasure to have you here. We've had the opportunity to interact here and there in the past, back when we were able to encounter each other stochastically or serendipitously. It's great to have you here on a more formal occasion, and I am, and I'm sure the public is really looking forward to your presentation today. So I'd like to suggest that you share your screen, and the stage is all yours.
Okay. I don't want to share the screen.
Scroll over your...
No, I know I have it, but it doesn't want to. The share button doesn't come out. So let me try to do something else. Okay, yes. That's what I've done. Oh, God, sorry.
One thing you can try if it's...
Yeah, no, I know.
You have it? It's all good? Okay.
Yeah.
Perfect. Yeah.
So, sorry, I'm like to be an expert in Zoom, and actually we have been Zooming for the last year extensively. Anyway, good afternoon, good morning, whatever, wherever you are. So I'm going to tell you today what we do in my lab in terms of understanding how one generates neural diversity. And so I'm basically a developmental neurobiologist who cares very much about how we build the brain, a bit how the brain functions, but mostly I believe that by understanding how the brain is built, we can understand how basically the logic of how it's going to function. And so as you know, the brain is made of many, many neurons, and there is a lot of diversity in those neurons. Again, starting with Cajal, we have been documenting this type of diversity, and by far it's the most diverse organ in our body, and we want to understand how this diversity is being established. Okay, so I mean, Denis told you that actually I was quite interested in stochastic fate determination, for example, in the photoreceptor in the eye. But what I'm going to tell you today is really exactly the opposite, in the sense that what I'm going to tell you is that the development of the visual system of the fruit fly is something which is very deterministic, which means everything is basically lined up from the genes, and there is no variation to that. Everything is being done very, very robustly and deterministically. Okay, so we focus indeed on the visual system of the fruit flies, because if you want to work on human brain, it's kind of a daunting task, because you have to 80 billion neurons, each one connected to 100 to 10,000 other neurons, and there is many neural cell types. I don't think we know how many cell types there is in the human brain. In contrast, the visual system, the optic lobe of flies, has only 60,000 neurons, which means out of the 100,000 neurons of the fruit flies, only 2/3 are dedicated to the visual system. The fly is a very visual animal, and this visual system has been characterized in exquisite detail, again, starting with Cajal, who spent a lot of time developing that, and Fischbach. And we know that is about 200 neuron types, and maybe we are missing a few, but overall, we know most of them. And of course, because of Janelia effort, there has been an EM reconstruction of those neurons, which means we know whose neurons talk to whom, who's connecting to what, and therefore there is a great advantage for them. Yet, this very simple brain is highly performing. The flies can fly at high speed without banging in any object, do some very complex looping. And of course, this is in the context of an animal when you can do very easy genetics and now single-cell genetics. And again, just to give an idea of how this looks like, again, this is a picture taken from reconstruction from the 80s from Fischbach, done actually with mostly gorgeous stainings. And you can see here that we have all those neurons that connect in between the different layers of the optic lobe. And I will tell you exactly how this happens. Okay, so the optic lobe is made.
Sorry to interrupt. Can you click on the little thing hide there, so that little box would go away?
Yeah. Sorry.
Thanks.
Okay, so the visual system is made of different layers, and you're going to see what some of the advantage of this visual system are. So first, there is of course the retina, and the retina is very simple. There is about 800 unit eyes. Each one detects 1 pixel. And the beauty of the system is 800 pixels are going to be processed individually through circuits that are going to be reproduced through a retinotopic map in the different sublayers of this visual system. First, mostly motion vision is processed by the 800 cartridge of the lamina. And then, and you can see the photoreceptor in green that project to the lamina. Those are involved in motion vision. Other photoreceptor, those here that you are in green, also project to the medulla. Those are those involved in color vision. And then there is a relay where basically from the lamina to the medulla, you have lamina cells that actually are going to do further processing of the motion in the medulla in a higher processing center. So the beauty of the system is that you have basically 800 circuits running in parallel to one another, and they're all basically almost identical to one another. There's almost no difference. Every part of the eye is processed very similarly. Of course, there are always some exceptions that confirm the rule, but for what we want to do, it's kind of useful to do that. So the beauty of the system is actually we have multiple times the same thing, and for us who are interested in lineage, it's a great advantage because we can actually do everything done multiple times in the system. Okay, so the eye is fairly simple, only 20 cell types, mostly photoreceptor and support cells. The lamina is very simple, only 5 cell types involved in motion of a black over white or white over black. Very simple system, but the medulla is by far the most complex, and I will spend most of my time trying to understand how the medulla functions. And then they project to the lobula, And the lobula is basically what is going to take the information from this retinotopic information from the medulla and then process the different visual features. It's a bit like the retinal ganglion cell in the mouse retina. They're going to separate basically what is motion, what is color, what is small object detection, what is looming, and all of this is going to be processed independently from the lamina neuron they project, the lobular neuron they project into some part of the central brain. Okay. So let me tell you first what the type of neuron that we have. Again, so as you could see here, that you can see that you have the different structure. And I didn't tell you that, but in blue, what you have is actually the cell bodies, and the cell bodies are completely separated from the projections, axon and dendrite, which are in the red region here. So if you focus, for example, on this part, you can see there's this 800 cartridge, okay? And you have a bunch of neurons, about 40,000 neurons, which are here. And those 40,000 neurons, if you divide by 800, it means that there is about 50 to 100 neurons surrounding each one of the single unit. And you have about 800 of those units running in parallel to one another. So what type of neuron do we have? So what we can do is do some sophisticated Markham experiment, which is single neuron labeling experiment. Take one neuron here and see what it does. Here, what you have one neuron. In red is a projection of the photoreceptor. So basically, we are right here, okay, in this region here. And so what you can see here is that these neurons, the fly neuron are monopolar, which means they have a long process, dendritic arborization, and then the axon is going to send information there. It collects information from the photoreceptors. And you can see this neuron is what we call unicolumnar because there is one for each one of the cartridge, each one of the unit, which means you imagine they're going to have 800 of this type of neuron. Okay. Then you have neurons which are a bit more complicated, which are multicolumnar, which means they cover a broader receptor field. This one, for example, will cover about 10 cartridge, which means if this one tiles is going to have quite a lot fewer neurons and maybe 75 neurons that cover the whole retinotopic map. Okay, and so we can do that over and over again. Again, we and Fischbach and Caral before and Janelia have done a characterization of those neurons, and now we have a pretty good idea about what kind of neuron here. So if we focus on the whole optic lobe, there are 200 neurons. Just in the medulla, there is 100 neurons, 100 type of neurons, and again, each one will cover basically will be there present in each one of the cartridge. And you have different type of neuron. You have first the projection neuron, which take information from the photoreceptor and send it to higher processing center. And you have the local interneuron, which are just locally, they don't send projection neurons, they just process information for whatever processing. But more importantly for what we want to say today is there is a unicolumnar neurons, which are one for each one of the cartridge. And we believe there is 25 to 30 type of those. And you have the multicolumnar neurons, which are much more complex. They vary to a large extent in numbers, and there is at least about 70 of those cell types that are occupying each one of the cartridge. Okay, so now we want to figure out exactly how this happens, and maybe first try to characterize the diversities that you have here beyond simply the morphology that you have described here. Okay, so first thing that we did as fly people is that we took advantage of lines which are labeling specifically one neural cell types. Again, the effort of Jerry Rubin at Janelia, when he started Janelia 10, 15 years ago, they basically made a lot of Gulf4 lines that where basically they took fragments of genes and let them express in the brain, and they noticed those that were expressed only in a very few, or sometimes like this one, only in one cell type. So you can see this Gulf4 is expressed only in one cell type, expressing GFP there. And therefore, what you can do, you can just dissociate this brain, fax sort those neurons, and try to get to their transcriptome. So we did that, and Janelia did that also. It's a bit of a very painful system because it takes a long time, and you have to go one by one. And basically, altogether, we have about 15 neurons which have been characterized that way, neurons for which we have a Gulf4 line, and we know exactly what their transcriptome is. Okay. But again, we have to go a lot further. It's actually encompass not only the medullary neuron, but actually all of the 200 visual system neurons. And so we are 50, we are far away from the whole description of all the neurons that exist in the medulla. And so this is actually 3-4 years ago is a time when a single cell sequencing came out of the market, and we saw the technique was absolutely appropriate for us. Actually, it was designed for us almost in a sense that if you want to investigate neural diversity, single cell sequencing is amazing for that. So a student, Felix Simon, and Nechel Osset, a postdoc, got together and start sequencing the single cell. And what you have here is actually a single cell sequencing of all the neurons from the adult visual system. So again, the adult visual system is supposed to be composed of about 200 neurons based on morphology. And you can see here what the beauty of the system is actually you can form those clusters, and those clusters are very, very well separated from one another. They have very distinct identity, and it's really great because now we can actually put separate, and we know we can describe the transcriptome of almost all of the neurons in the optic lobe. In fact, out of 110,000 cells, like it was published just a few last week, we got 193 clusters, and among those, most of them are neurons, which means we have almost all of the neurons that compose the optic lobe of the fruit flies in the adult, okay? And among those, through the works that I described to you earlier, and also through our own identification of individual cell one by one, we can put a name on many of those clusters that we have identified. Some of them remain cluster, which means just transcriptome without assigning name for that. But we are working on trying to make sure that we identify all of those clusters. Okay, so again, what you can see here also, there is something striking that the size of the cluster does vary quite extensively. You have a group of very large clusters, okay, they're almost all about the same size, and I will come back to this one later, and then a large number of smaller groups of clusters. And I think if you remember what I told you earlier, you have two types of neuron. You have the unique columnar neurons, which are present in every cartridge, and those are going to be those which are forming big cluster because each one of them is present 800 times in each hemibrain of the fly. And then the smaller cluster are going to correspond to the multicolumnar neurons. Those are present in smaller number because they have a much larger receptor field. And if you put a name now, the neurons, exactly what you see here. The large cluster has the unicolumnar neuron. The small cluster actually is the multicolumnar neurons, and you have some glial cell also. So again, now, and again, we are working to identifying all the missing cluster. Most of the large cluster have been identified, but we're trying to figure out exactly what is a small cluster because we want to have an exhaustive description of all the transcriptome of all the neurons and try to connect this to exactly the function, the connectivity, and the function of those neurons. Now, when we got this result, something that did bother us is actually this very large cluster. And this very large cluster, usually in single cell sequencing, when you get a cluster in the middle, is actually crap. This is not crap at all. It's actually the most important neuron in the visual system. The T4 and T5 neurons, actually 8 neuron types, T4 and T5, 44 and 45, which are involved in the detection of motion. T4 are involved in detection of the motion of a black, of a bright on a white background, and the opposite for the T5. And A, B, C, and D subtype just measure motion along the front to back, back to front, up and down. The fly is a flying animal, and therefore it needs all 4 cardinal directions to do that. And yet we know there are 8 neurons there, and they still cluster into one single cluster. And we thought about it, and we said, okay, maybe this is because we look in the adult. In the adult, indeed, those neurons are very, very similar to one another because they measure the motion of an object between 2 points in space next to each other. And they compare basically 2 points in space. And the function is the same. What matters is that how they connect those 2 points in space. Either they measure 2 points, one next to each other on the anterior posterior axis, on the dorsoventral axis, or vice versa. And therefore, we thought if we can look at connectivity, we might be able to see difference in those neurons. And therefore, looking in the adult might not be the optimal thing to identify neural diversity. And indeed, maybe what happens is when you establish connectivity is where the maximum neural diversity will be able to be detected. And so what we did is say, now we have the adult transcriptome, we know all of those neurons. Let's see what happened during the time those neurons do develop. Okay. So, and before we go on, I mean, we still need to identify many of the clusters which are missing. And what you have here along the x-axis is basically the number of clusters, and on the y-axis, actually genes, many transcription factors often that specify each one of the neurons. Okay, and therefore we know many of those have been identified, but we can actually find genes which are often expressed in one or very few clusters and try to see where in which neurons this gene is expressed. And again, thanks to the tools that exist in fruit flies, we can actually take a gene and figure out exactly in which neuron is being expressed. And so we're very busy trying to do that. It takes a lot of time, but we are progressing quite well. Okay. So again, going back to the system, what happened, for example, to those neurons during development? Are we able to distinguish the 8 subtypes that are forming those neurons? So we say, okay, the problem is that doing single cell sequencing in the developing brain or optic lobe It's nice, you can do it, but you cannot identify the cluster because the neurons at this stage have very little form and shapes, and we don't know very much about that. Almost nobody has ever investigated it. But we thought maybe if we start from the adult and then start a bit earlier and earlier and earlier and earlier, we might be able to match, for example, the neurons that exist at P70 with the adult neuron, and then those at P50, those at P70, P50 to P40, and then walk back towards the early developmental stages and try to see whether we can match the transcriptome of those neurons earlier and earlier in development. And so that's what Neuchâtel did. It worked beautifully well. What he did is he did a single cell sequencing clustered at P70. And then what he did identification, ID transfer from the adult to the P70. And actually it worked beautifully well, perfectly well, which means all the neurons in the adult could be matched to the neuron at P70. And then from P70 it could move to P50, and then to P40, P30, and P15, which is basically the stage, basically almost a percentage of developmental stages. Okay, so basically what we have now is that the development of all neurons, almost all neurons in the optic lobe from the time almost they were born, or this, I mean, basically a few hours after they were born, all the way to the adult. Okay, so a lot of interesting things you can see here. First, you can see that these clusters are forming very well there. But then when you go earlier and earlier, you can see the clusters start to be elongated, often merging with one another, which I guess represents the fact that those neurons are just in a process of maturation. So these long tails are maturating process, and I will go back to this a bit later in my talk. Okay. But so now the question, you remember, we did that because we thought that those T45 neurons, those 8 subtypes, which we cannot distinguish in the adult, might be distinguishable earlier in development. So now if you look at P50, so at mid-development of those neurons, you can see now that exactly what happened. Now you see 8 beautiful clusters corresponding to 4 T4 neurons and 4 T5 neurons, and those correspond to really the diversities we are looking for. So which means that, doing an exhaustive single-cell transcriptome of adult neurons might be a way to get to neural diversity, but the only way to get full neural diversity will be to look at during development when this diversity is being generated, because often neurons which are connected differently can function the exact same way, can have the very same machinery for functioning. So it's one of the things that we have now. We have the developmental transcriptome for all neurons throughout development. Again, there is a lot of things we can do, and of course we are very busy exploiting all of this data, and I will just to give you a few things there about what we can find. And again, there is a lot more, and if you're interested, again, the paper came out last week in the issue of Nature, and you can look into it. It's a fairly heavy paper, but it should be interesting for you to read if you're interested in the process. Okay? So first thing we find is actually, I told you we have identification, ID transfer from the adult to the earliest time point. And what we find actually, some neurons actually exist during early time point, but we don't find them in the adult. They don't exist in the adult. For example, these 2 clusters here exist earlier, they exist at a later development, but we cannot find them in the adult. Okay, so we say, what are those neurons? Okay, and if you try to find marker for that and try to see whether we can mark them, we can find a fairly large number of neurons that actually are very bizarre because they are flanking on the dorsal and ventral aspect of the optic lobe, and they just wrap the neuron, but they never penetrate deep inside the optic lobe. I mean, there is nothing of those projections from those neurons inside. Okay, so we said, what are those cells, and why don't we find them in the adult? So again, you can see them during development. Early in development, you can see those neurons are labeled with some specific GAL4 marker, and again, you can see they wrap without penetrating inside the optic lobe. And then later in development at P85, just basically a few hours before the flight is going to close, you can see those neurons start expressing caspase, which means they start to die, which might need to explain why you don't find them in the adult, because they are dying at late in development, and indeed in the adult, with the same marker, you don't find them. Just to test whether indeed this is dying, what we can do is label them with a marker that actually prevents cell death. This is a P35 molecule. And if you prevent that death, you can see those neurons survive, and you can see them hanging around in the adult. And so we try to figure out what those neurons are doing. And is it necessary for them to die? What do they do during development? We don't yet know. And to some extent, for the vertebrate people, this kind of resembles the chiral reticule cells that exist in the layer 1 of the cortex and have a role in development, but they're not necessary in the adult. And so we have a lot more things to do and to achieve to be able to understand all those processes, all these neurons, all this development. And again, I don't have time to tell you all of this, but again, you can look in the paper if you can. Okay. Okay, so now what we have, I gave you a fairly good idea about what happened in terms of neural diversity, and again, how those neurons differ from one another, and why they have actually we have a deep understanding of the transcriptome of those neurons. But the question is, how is this neural diversity generated? Okay. So how do those 200 types of neurons get produced? I'm going to focus on half of the neurons, those that are in the medulla part of the optic lobe, because it's where we know the process best. So 100 neurons are coming here. They are making 100 neuron types. They form 40,000 neurons again, and there's about 100 cell types. Each this 100 cell type surround each one of the cartridge, either as unicolumnar neuron or a multicolumnar neuron. So can we understand how those 100 neurons are being produced that surround each one of the cartridge? So I'm going to make a very gross exaggeration here. What I'm going to tell you is that actually there are 800 neuroblasts, neurostem cells, and each one of them produce most of the neurons that surround each one of the cartridge. It's not exactly true, but overall the concept is there, which means that many of the neurons that are surrounding one cartridge appear to be generated by one neuroblast. Okay, so that means that if it's the case, that means that each neuroblast must produce a large number of different neurons. One neuroblast, maybe 50 to 100 neurons. Okay, it's not 100, but it's not so far from there. Okay, so again, this is the 40,000 neurons that compose the medulla. The neuron, the cell body are all here, and each one they make 100 cell types that surround each one of the cartridges that you have here. Okay. Now, if you know something about fly development, you know there is something striking about the way the fly developed, and this was starting again from the people who studied the eye in the 90s, is actually the eye of the fly, just the place where the photoreceptors are, developed in a sequential manner with a wave of differentiation. What you have a monoepithelium here, the red part here, early in development, and what's going to happen, they're going to wave of differentiation, they go from posterior to anterior, where you get to recruit photoreceptor, and the wave will recruit more and more and more photoreceptor, which means over a period of 3 to 4 days, they're going to form an eye, and they're going to form photoreceptor, and the fly photoreceptor are in fact neurons, which are going to have photoreceptors that project their axon towards the optic lobe over a period of 3 or 4 days. And they're going to have to meet their target neuron in the optic lobe to be able to connect. And again, this is going to happen over a period of a few days. In fact, the very same thing happens in the brain, in the optic lobe, and in particular, there is a neuroepithelium here in purple, and you can see the columnar organization here. The neuroepithelium that are going to form this medulla structure, which I just talked to you about. And this neuroepithelium, like the neuroepithelium of the fruit flies, will also undergo a wave differentiation, whereby a wave of differentiation will go and cross the neuroepithelium of the medulla, and it's going to go that way, cross that way, and in this wake, they're going to produce neural stem cells called neuroblast, okay, which are going to be produced again over a period of 3 or 4 days. So how does this happen? So again, the neuroepithelium is this crescent shape here, structure here, and at the edge, at the outer edge of this crescent, you're going to have the neuroepithelium being transformed into neuroblast, neural stem cells. And you can see here a picture from Tetsuo Yazuki. What you can see here is the neuroblast at the edge, and sorry, they're going to become, their stem cells are going to start producing neurons. And a few hours later, the wave is going to progress from this outer edge of the crescent towards the inner edge of the crescent and produce a more neuroblast in a progressive manner over a period of days. So in a schematic manner, what you have, so neuroepithelium is here, the first layer of neuroblast form. They don't delaminate, it's just a transformation of the neuroepithelium into neuroblast. And then a few minutes later, another neuroblast will form, and so on and so on and so on. Okay? And at the same time, in black here as a photoreceptor, which also comes sequentially from the sequential development of photoreceptor in the neuroepithelium from the imaginal disc of the eye. And so they're coming also. But you can see here, the neuroblasts here are forming here, and the neuron arrive here, and they're far away from each other. Ignore the yellow part, which is it's the laminar part of the optic lobe, which is depends on the photoreceptor. But for sure, this wave of differentiation here is far away from the connection from the photoreceptor. So let's try to see now, if you make a cross-section through this structure and flip for 90-degree angle, what you can see here, cross-section, again, the neuroepithelium is here, okay? And the neuroepithelium is here. And then here on the outer edge, this neuroepithelium will become a neuroblast. And neuroblasts do what neuroblasts do. They, like stem cells do, they divide, renew themselves, and produce a cell, which actually there is one intermediate precursor cell called a GMC, which divides only one single time to produce two neurons, one notch on, one notch off neuron. Okay? So first division, you produce two neurons. And then a few minutes later, an hour later, the next neuroblast will form from the neuroepithelium, form a GMC, and you're going to form two neurons. But this one keeps dividing, and then it's going to progress like that. The wave will progress, and at the end, what you're going to have, because it's so packed, you have long columns of neurons that are the product of a single neuroblast. And again, I will try to argue that most of those neurons here that are produced, each one is different from the other, and they're going to compose most of the neurons that surround each one of the cartridge of the neuroepithelium. Okay? Okay, to show how this happens, you can see here, so you have young neuroblasts that just formed from the neuroepithelium. These are all neuroblasts that formed like 2 days ago. And of course, these are just young neurons that just formed from this neuroblast, and those from a few days later when the neuroblast used to be young. Okay? So you have a temporal axis, and actually you have two temporal axes, the age of the neuroblast and then the age of the neuron. And I will try to give you some information about that. So if you do a labeling and you label only a single neuroblast, and you can do that in flies, you can see beautifully this column of neurons, of two neurons because of the GMC dividing on, notch off neurons. You can see these columns of neurons that are going to be forming, this long line of neurons, which are the progeny of those. And again, you're going to see that each one of these neurons are all different from one another, and they all perform different functions. And now, indeed, now the neurons are forming. Now they're going to meet the photoreceptors that come from the eye here in a sequential manner, and here they're going to make connection. And we are very busy trying to figure out how indeed they find each other, and also how this connectivity occurs in a synchronous manner in such a way that the speed of motion of the morphogenetic furrow in the eye match the speed of the progression of the neurogenic wave in the neuroepithelium of the medulla. Okay, so let's try to focus our attention on one of those neuroblasts here and to see how this neuroblast could produce a large variety of neurons. Okay, so we are not actually in a conceptual blank, because actually a few years, many years ago, 2 decades ago, Chris Doe in Oregon has actually looked at the neuroblast, the neural stem cell from the flies in the ventral nerve cord. Okay, and what he defined actually is that actually there is what we call a temporal series of transcription factors. How does it work? Simply, the neuroblasts, when they are born, they express a transcription factor called hunchback. And when they divide, they renew themselves and produce a neuron that expresses hunchback. But then the neuroblast gets tired of expressing hunchback, is going to switch off hunchback and switch on cripple. And next time it divides, it's going to produce a cripple neuron. Then it's going to switch on to PDM, produce a PDM neuron, and then a castor, produce a castor neuron. Okay? So beautiful conceptual things. And of course, people got very excited 20 years ago when they found that, because they say it's exactly what we see in many of the systems when you have these layers, this production of different layers. And of course, I should remind you of the production of the cortical layers of the mouse or human cortex, which is exactly that. The same radial glial cell is going to produce layer 6, layer 5, layer 4, layer 3, and it will be perfectly well adapted to that. In fact, it has been reported that hunchback equivalent in vertebrates seem to be specifying the deeper layer of the cortex. Okay. So people just jump on that and try to find again whether hunchback was expressed in the visual cortex, in a cortex of the mouse, or in a retina of the mouse. And they did find, Michel Cayouet did find hunchback, PDM, and castor in the mouse retina, but otherwise, besides that, there was very, very few reports. And again, Denis is actually working on this, and there's a beautiful paper also coming out from Jim Briscoe, where he looked at in the nerve cord of a mouse. And but so far, there is no real very good series of transcription factors like this one that exists. But we say, will be great in our system, because since we have this sequential recruitment, this sequential production of neuron, you have a young neuroblast and you have all neuroblasts. You could imagine that you're going to have hunchback, cripple, PDM, castor. We should be able to visualize this in our system in one shot, in one eye, in one snapshot. Unfortunately, none of those genes was expressed there, so we got to be disappointed. But we got actually lucky, because in fact, we got the very same thing, except we had to change the name and we extend the length of it. That means, in fact, the very same concept that Chris Doe put forward a few years ago is true in our system, except we have to change the name of the transcription factor. Young neuroblasts express homothorax, clumphus, homobrain, ILS, repaired, dactyl, and I will give you a few more later on in my talk. Okay, so it seems that indeed the way the neural diversity being produced, that this neuroblast here, this old neuroblast, when he was young, he did express homothorax and produce homothorax neurons. Then it's through to clump then to homeobrain and ALS. And you can see now how this single neuroblast can produce indeed a diverse array of neurons. Okay, of course, if I told you we want to produce 100 neurons, this is a bit short of 100, but there are ways around that I will not have time to tell you about, where the combination of those transcription factors and also diversification where the neuroblast is located in the neuroepithelium will actually increase the diversity, and actually we can account almost for all of those 100 neurons coming from this type of temporal series, which again I'm going to tell you later will be extended a little bit. Okay, so what happened? Young neuroblast express homothorax, ALS, repair DACIT. How does the clock tick? How does the neuroblast decide to switch from one phase to another? So what we can do, again, do experiment. We can take a single neuroblast and mutate, for example, the gene stop paired and see what happened. And if you do that, actually, you have a very nice result in the sense that the temporal series continues. The neuron keeps dividing, but it gets stuck into the previous temporal window. Okay, so if you remove stop paired, you get stuck into ALS, never progress further. Which means this gene ALS most likely is important for specifying the fate of the neurons that come from the ALS temporal window. It's also required for the progression of the temporal series in this series. Okay, so the idea will be, and actually seems to be true, again, every rule has many exceptions, and nature loves exception, is that the gene X, in this case stop paired, is required for repressing the gene before it. Is why if you don't have stop paired, ALS gets maintained. It's also required for activating the next gene DACIT. Okay, and is why if you don't have stop paired, you don't activate DACIT and you get stuck into the ALS temporal window. So it seems that the progression of the temporal series is simply a progression through activation repression. And it's so interesting, and actually I was discussing this with Denis just before the talk, that this is something that each transition from one neuroblast stage to another temporal window takes a few hours, maybe an hour and a half. And if you do the same thing in mouse, it takes a day. If you do something in human, it takes a week. So it's possible that actually a transcriptional cascade might not work with this very long timeline because it gets too slow to do that. And so that means we have to invent some other system. And I don't know what those other systems are. Okay, so the end product is this picture that I really love. It's actually the picture of the neuroepithelium, which is here. That's a young neuroblast that just formed. The neuroblast that formed. And then the age and that's the older neuroblast that went, they used to express this gene, now they express this gene. This is ALS, repaired, DACIT, TLS. And again, this is only 4 of those, but you can have many more of those. Okay. So these are pictures that you have in the larva during the development of the system. And so this is now going to tell you something is a big advantage, a disadvantage. The advantage is actually if you have one snapshot at this stage that I showed you here, you have all neuroblasts at all stages of development. All neuroblasts, young neuroblasts. And presumably, if you look underneath, those neuroblasts are producing neurons, which are also going to be at different stage of development. It means those produce the neurons, the young neurons. That means those neurons express homothorax. They're going to produce many neurons, including earlier those express homothorax, so repaired DACIT, TLS, and now they're producing the late-born neurons. Okay? So that means that you have all the time point, but if you want to do single-cell sequencing on that, you also have to realize that there is a lot of diversity, because now it's not only diversity of neuron, but also diversity of the stage of development of those neuroblasts, and of course of the neurons that emerge from them. So Nikos Konstantinides, who is going to be a faculty at Institut Jacques Monod soon in Paris, did the single-cell sequencing of the optic lobe at a larval stage. And this is quite different from what we saw before when you have this beautiful cluster that you could see in the adult. Here, instead, it's a very elongated system whereby, it's much harder to imagine what's going on there. You have these long lines of things, and presumably some of those lines might represent time, or at least pseudo-time. And so I want to try to explain to you what those lines are and what we can extract from this information. Okay. So first things, the neuroblasts. We have marker for neuroblasts, and you can see the neuroblasts are mostly from this bunch here. Okay. And these actually the neuroblasts I just described to you. Those that go to this temporal window, they are producing the medulla. This is actually the neuron from the medulla. Here, actually, there's other neuroblasts. There are neuroblasts that from other parts of the optic lobe that it dissected, for example, the lamina or the lobula or the central brain. So I'm going to ignore that for now, but I want to focus my attention on what happened here, how those neuroblasts that are here are going to produce GMC, those intermediate precursor cells, and then they're going to produce neuron in gray. Okay, so neuroblast, GMC, and neurons. And again, the question we asked is how is the continuity? What the pseudotimes they go from the neuroblast to the GMC through each one of the mature neurons. So what we can do is now try to integrate this data set, this information in a larvae, which is very elongated, very developmental, with the information we got from the single-cell sequencing at the earliest time point that Neshat was describing, which is about 15% pupation, 15% of development. Okay, and basically what you have in blue that you add on the same data set, you ask that the P15, the data of the young neuron, which are already produced but are in a process of maturation, and you can see actually that you can put those neurons at the end of those long trajectories. Okay, so basically what you can see, you can almost follow from the neuroblast all the way to the mature neurons and to see what happens here. Again, I'm not talking here about lineage. Those lines are not lineage, they're just maturation of neurons from the young neuron coming from the neuroblast all the way to the mature neuron, which are here. Okay, and I want to draw your attention on one neuron. I will focus my attention more on this MI1 neurons. Okay, it's not connected to any good neuroblast, but as you can see the mature neuron here, and actually you have the developing MI1 neuron here. Okay, so now what we have a series from neuroblast all the way to neurons, real neurons, form neuron, maturing neuron, and the question is that can we take advantage of that to understand more about the process? And the first thing I want to do is to focus my attention on those neuroblasts which are here. Those, you remember, they were labeled by the marker of neuroblasts, and those are neuroblasts that are forming all the neurons of the medulla, which is here. Okay? And you see that kind of structure in this UMAP structure, this UMAP plot, that kind of structure of these layers. And actually, the amazing thing is that if you now try to place those temporal transcription factors, which are described to you, they actually amazingly, they almost look at in situ hybridization that you see. But it's actually the UMAP plot, so it's not just completely fake. It's just bioinformatics. And you can see that if I flip between the different images, you can see you have homothorax at the bottom, eyeless, two paired dactyl, which means that you have, if I go there, you have a temporal axis, which is a maturation of the neuron here, the maturation of the neuron cell. But then if you look along this axis here, you actually the temporal series of the different neuroblasts. Okay, so again, I always find this picture totally striking that you can see on the UMAP plot the pattern that you see really in a maturing neuron. Okay. So now when we have this neuron in hand, what you can see is that you have definitely a temporal series coming from here to here, young neuroblast or neuroblast. Can we now identify other of those temporal transcription factors which were not identified? I didn't tell you that, but the way we discovered those homothorax ILS without dactyl was a very haphazard way, which means we did just basically the antibodies we thought might be useful, all the collection we had. We worked, as Denis told you, in the early embryo, which means we had a lot of antibodies from early embryo. So we tried randomly, and this is actually just not a very well-designed screen. It's just the product of luck, chance, and guess. Okay? So now can we do something much more systematic and to see whether we can find genes, transcription factor, which express peak at some point and then go down, peak at some time and go down, and so on. Okay, and we know this might be the case because those that we know do exactly do that. Okay, so Nikos did that, and exactly what you have here again. Yellow is higher expression pattern, and you can see that as you go from early to late. So for again, this purely pseudo time, you can see early you first start with genes which are involved in the transformation from neuroepithelium into neuroblast, and then you go to homothorax, ILS, homobrains, to be paired, dactyl, and teles. And then you can add a few more in between because you can see so as a gene that peaks and go down, peaks and go down, peaks and go down, peaks and go down. This one actually has 2 peaks. And therefore now we can expand the series from 5 or 6 transcription factor, temporal transcription factor, to actually a dozen transcription factor. And basically this allowed us to basically be exhaustive. We believe we do have the whole temporal series of transcription factors that exist in this neuroblast of the medulla part of the optic lobe. And so basically what happens is that the neuroblasts, when they are born, they express homothorax, then they go to ERM and to ILS. And what you can see is that the rule that defined earlier that gene X activate gene X plus 1 and repress gene X minus 1 seem to apply to most of them, but not all. Some of them bypass some of the steps. So it's always, the diversity that you see here. So again, we believe that all those genes here, those temporal genes, are all involved in the progression of the temporal series, and most likely they're also involved in a specification of the neuron that are born from this the stage of development. Okay. So this is great, and I think we do now believe have exhausting series of transcription factor, and we are very busy trying to basically establish how those work, how they function, how they regulate each other, and what they do in terms of the activation of the what Oliver Huber called the terminal selector genes. And we do believe we have this terminal selector gene. So again, these are the transcription factor in the neuroblast, the temporal transcription factor in the neuroblast. And if you look at the neurons which are produced by those neuroblasts, so neurons actually below the screen, you can see here those selector genes. What you can see here, the neurons are born early express BSH, so they're born later are called RAND, drifter, toad, and stopper, and you have quite a lot more. These are genes in neurons. These are neurons, they're not neuroblasts anymore. These are genes in neurons, which I believe could be those terminal selector gene a la Oliver Huber that actually mark the different neurons born at different stage of development. And so we have now a complete series not only of the temporal transcription factor, but also of all the genes that appear to be produced by this transcription factor. And plus, we can just exploit the transcriptome from the single cell sequencing to figure out exactly what are the genes that express in all those different neurons and how they get them. So we believe that most of those genes actually are expressed very early and they're controlled by the temporal transcription factor and they're maintained all the way to adulthood, which means they really fit the bill of the terminal selector gene. They're going to basically specify all the features of a neuron, its shape, its projection, its connectivity, its neurotransmitter receptor. Okay? And so we have all the tools here to investigate it, not for 1 neuron, not for 10 neurons, but for 200 neurons. Okay? So again, this is reminiscent to what we see in a visual, in a cortex of a mouse. Again, you remember those layers 6, 5, 4, 3, 2. Again, same type of diversity. Those two pictures are very similar to one another, very similar. And the question that we know also is that those radial glial cells that are produced here are going to produce layer 6, layer 5, layer 4, layer 3. And those beautiful clones from Songhai Shai and Simon Ippenmeyer show that indeed, if you label one single radial glial cell, it's going to produce a neuron layer 6, 5, 4, 3, 2, sometimes produce 2 neurons, but overall that means that each of the radial glia does follow exact same system as what I described to you, except much simpler, but is going to produce neuron in each one of the layers in a sequential manner. Now, how does this happen? Is there a temporal series of transcription factors there? I don't know, and I will be very anxious to figure out. Although we exploited some of the data that Denis has produced, and we cannot find really genes that go up and down. But again, James Briscoe has a paper just coming out where he described those kind of things where at least the temporal factor are expressed in neuron of the ventral nerve or the dorsal nerve cord, and again we don't know yet what they are, but we'll be interested to figure out what's going on. Okay, so let me go back and try to give you a last bit of information on once the neuroblasts have produced neurons, okay, and you can see those long lines. What happened along these long lines of differentiation? And I'm going to focus my attention again on the MI1 neurons because you can see beautifully well the progression from neurons that just were born to neurons that are getting mature here. Okay, so we can actually do some pseudo time, and we can now integrate not only these pictures that I show you here, this picture here between P0, the larvae, and the P15, which is 15% development. And you can even add the later stage of development, and you can draw a trajectory, and you can see what are the genes that express there in those MI1 neurons. And actually, I will generalize that to all of the neurons we have been able to look at, and here is the result. So what you can see, not surprisingly, the first genes that come out actually are DNA replication because these are just, the neuroblast divide, and all the GMC, the intermediate precursor divide. So we have DNA replication, and those, and then the next gene to come out actually are basically translation gene involved in translation, ribosomal assembly. It's not very difficult to imagine why, because the neuroblast divide in a very asymmetric manner. Again, quite different from the radial glia. The neuroblast remains very large. The neuron is pretty small. So if it's going to send some axon, dendrite, it needs to basically grow quite tremendously to be able to have a size enough so you get enough ribosome to be able to produce that. And then as a neuron develops, of course, we're going to get dendrite development and axon guidance. So it means the neurons are going to form the dendrite, are going to send the axon and get some guidance to be able to find the target. And again, this is basically as a neuron matures. But then there is something quite surprising that comes up. Exactly those two types of things: the regulation of membrane potential and the chemical synaptic, which means neurotransmitter gene appear very early in development. They are just born like a few hours ago, and they already express the genes that encode, chat and VGLUT and whatsoever. The genes that are going to be required for the formation, the production of neurotransmitter. And of course, these are going to be held at fairly high level, and then later on, they're going to peak when the neuron mature, and they're going to start using their neurotransmitter and their neurotransmitter receptor. But really, there's a huge increase there in which we do not understand why this comes up so early. Now, to just dim a bit this statement, in fact, indeed, those genes are expressed, the RNA are expressed. But if you look at, so you can see here exactly those are in this diagrams. You can see that the GAD and the VGLUT and the chat neurons, they all kind of grouped together. It means they have some commonality in terms of where they are. So they express a neurotransmitter. They share between neurons that come from the same origin. Okay, but even though they express very early in the development, you see these are far from being mature neurons. The mature neuron will be at the tip here. But if you look at the brain, exactly the larval part where the neurotransmitter are expressed, because indeed there's a functioning neuron, this is the part we are looking at, which is developing in the larvae, and basically they're not functioning neurons. And you can see actually, although you have chat and GLUT expressed there, the protein is not expressed there. So most likely what happened, actually, the neuron express the genes, but the genes are not translated for some reason, which we need to figure out at some point. So basically what I just told you today is that what we have is that we can define different temporal axes of development, a temporal axis along the neuroblast, along this axis here, a temporal axis along the maturation of the neurons. And of course, you can see that all those neuroblasts are going to basically follow those rules. And again, the question will be that, is there any input from, for example, expressing factor? And actually, we know that at about this stage of development, if the sort of time, ecdysone will be there and actually will induce some further development. So the idea will be that indeed, what you have, the temporal transcription factor turn on these selector genes in the different neurons, and those different selector genes will act at different time points depending on which expressing input they receive from the outside to produce, axon guidance or neurotransmitter or potassium channels. Okay, so again, we can see here, we can describe in exquisite detail what happened in those developing neurons from the time they were born as neuroblasts to always to the time when they're actually in adult. Okay, so I want to stop here and thank Nikos, who is basically the one who introduced us to single-cell sequencing. Again, he's moving to Institut Jacques Monod in sometime in the spring, and then he was relayed by Felix and Neshet, who are Felix is a student, Neshet is a postdoc, who actually have learned how to use that, and as they become a very good expert in bioinformatics, and they are very, very good. And then I have a lot of people in the lab who are doing something else, try to characterize those different types of neurons. And again, as Denis said, we also have a small project on different systems, different types of insects, including some other type of flies, and ants also that are doing collaboration with Daniel Randberg. Those are the people who left the lab to take on their job somewhere else. So I'm going to stop here and take questions, and thank you very much for your attention. Bye.
All right. Thanks a lot, Claude, for this beautiful overview, more than an overview, beautiful trip and detailed account of how this complex system emerges. You're back with us. You're seeing the screen.
Yeah,
yes. So while people in the public are setting up their questions, I have one. I mean, I have several, but I'll start with one. In the first, I guess it's a UMAP, the one where you're showing this nice individual clustering of different cell types in the retina, where they're so distinct from one another, where there's several hundred types of cell types in the adult.
Yeah, it was a Tisney.
It's a Tisney. Okay. I'm struck by two aspects. One of the first one you mentioned is the fact that they are so distinct, and of course the question that comes to my mind is the extent to which this is different from what one sees in mammals, right? Where it seems like we have this more of a bulky kind of cell types, and there might be intermediate cell types. Somebody in the labs calls this the gros pâté effect, where we have really a big chunk of cells that are thrown like that. So your speculations on this, on intermediate cell types.
Yeah.
And maybe the other one is, I'm struck that within one dimension or two dimension, these clusters are so distinct. It would suggest that what differentiates one cluster from the other is along these two main axes. You see what I mean? If you think of it in 3D, it's surprising that you would find an angle where they would all be so distinct from one another.
Okay, so to answer your question, again, I don't like the idea of a continuous, cell fate. I mean, to me, cell fates are cell fates. It means cell fate A and B and C. I don't think there is A prime or A 2/3 A and 1/3 B. I don't like this idea. It's just preconceived idea, my strong bias. It's possible that depending on when you look, you might have actually a neuron at different stage of maturation. And one of the beauty of the fly eye, the fly visual system, is actually, again, everything is very deterministic. There is very little place left to plasticity. I mean, to a larger extent. Again, we are working on a paper where wave of calcium can maybe modulate it. But overall, I think this is very, very deterministic, which means everything follows a very strict line. And again, everything is exactly the same because everything is so determined by those temporal transcription factors, by the genome. Those neurons are specified by the genome in a very, very strict manner. So it's why I think we also have a large number of neurons. And also, I think in mouse, what we all underestimate, there is no way if we have 200 neurons in the visual system of the fruit flies, we don't have many more in the mouse system. And actually, I was very struck by some of the. I mean, you remember when we were together in Beijing last November? George Shane was comparing the human and the mouse retina, and he found more diversity in the mouse retina than the human retina. And this is a bit surprising to me. And it seems maybe it's simply that the fact that yes, there's a very good explanation for that, but I think it might be due simply to technical problem. Maybe there is so much diversity that some of the cell types are being merged with each other. In our case, our cell type are limited number, and therefore each of the cluster we have is really a single cell type, and therefore it's not a gros pâté. It's actually a very strict, very limited number. Now, if you look in a smaller cluster, we do get some petit pâté. That means we do find some neurons that actually are most likely some clusters which are mostly made of a few 2 or 3 neurons, which actually now we are discovering. And actually, I didn't talk about that, but even in the gros pâté, in the large cluster, we find something interesting, and actually that some of those large clusters seem to be split into 2 subclusters. One has come from dorsal part of the optic lobe, facing dorsal part of the ventral part, and they're distinguished by the expression of 2 Wnt genes, Wnt4 in the ventral part, in the dorsal part, and Wnt10 in the ventral or whatever. Okay, so basically 2 subclusters, and if you look in detail, you can find they're slightly different based on this, and they cluster slightly. So a big and they're the very same cell types, but they are modulated by whether they look up and they look at the sky, whether they look down at the soil, at the ground, and indeed they're going to process information slightly differently. So it makes some sense in retrospect. But I think it's mostly the answer to your question is really because we have a limited number of cells and we have very deep single cell sequencing, and in proportion number of cells per cell type is actually higher than in most of the mouse experiment because the denominator is way too high in vertebrates.
Okay, thank you. I'm going to take a question from the public, from Sarah Palace. What is the role of positional information in setting up the transcription factor cascade, if any?
Okay, so very good question, and I sometimes talks about it. So from what I told you, it gives the impression that every one of the 800 neuroblasts I described goes to the exact same temporal series and forms the exact same neuron. First statement is correct. They all go to the very same temporal series. However, the statement they produce the same neuron is not true. Depending on where the neuroblasts are coming from, they're going to have the same temporal series. The same temporal series is going to produce different types of neurons. Let me explain that. In fact, each neuroblast produces 2 types of neurons. Neurons that care where they come from, and neurons that don't care where they come from. That means that if you have 800 neuroblasts, those neuroblasts produce neurons that are all the same among those 800 neuroblasts. Those are unique columnar neurons. You need 800 of them. But then each subset of the neuroepithelium produce neuroblasts, and those are going to produce neuroblasts. They're going to produce neurons Some of them will not care about where they come from. Some will care very much where they come from. They're going to produce in a smaller number. So the way you increase diversity is that from this spatial patterning of the neuroblast, depending where the neuroblast is going to produce both common neurons and neurons that are very specific to those different regions. Again, it's hard to explain without a diagram, but overall, basically, we believe that it's ignoring the spatial information is depending on a notch activity. So you remember the neuroblast divides, give us it produces a GMC. The GMC divides once more, notch on neuron, notch off neuron. After the neuron ignores the spatial information, the other half does care about the information and is going to produce different types of neuron. So it's a way to set up the stoichiometry of those multi-columnar neurons, because some are very low abundance or higher abundance, depending on the coverage of the receptor field.
I see. Okay. Thank you. A question from Shingo Nakazawa. Thank you for a wonderful talk. How do you think flies and mammals acquire a similar temporal neurogenesis mechanism? Convergent evolution, ? Mechanisms from the common descent, etc.
Okay, so it's a difficult question. But first, I don't know. I mean, I've not been able to figure out whether indeed there is something common between the vertebrate and the flies. Again, this temporal series is from Chris Doe, and Chris Doe has other temporal series in the central brain neuroblast or from us. Again, besides Michel Caillouet in a mouse retina, there is not much similarity. And again, in a paper which Nikos is trying to write right now, we took actually the data from Denise and tried to see whether we can do the same thing that we did in the neuroblast in the larval stage, and to see if we can find transcription factor, which in radial glial cell will go up and down. And we don't find that. We find one or two which are genes which are known to be involved in temporal patterning. That's the RNA binding protein, not DNA binding, RNA binding protein called IMP, and the other one is called CHINMO, it's a transcription factor. But it's one or two, and it's not a temporal series. So we don't know. And again, I was trying to argue maybe the time is what matters. Here it's too long to have this temporal series. I mean, Denise argues that there is maybe some external extrinsic cues like wind factors they can specify the neuron. I don't know. I'm pretty sure we're going to find them. And again, James Briscoe seems to do find them, but I'm not sure exactly how they function. And I think it's just that, it's a bit more complicated to work on mouse. And again, our great advantage is that we have on one time point every developmental stage, which we don't have in a mouse. You have to do it so it's much harder to do pseudo time in a mouse cortex than it is in a fly larvae. So maybe it's why we don't find them. But again, I'm at a loss. I cannot explain it. And if it were to be the same, for example, like in the case of Michel Caillouet, Hunchback, PDM, and Castor, I don't know. When this gene was first discovered, Chris Doe was arguing that Hunchback, Cripple, and PDM was actually gene expressed along the axis of the spatial axis of the fly embryo. Hunchback is anterior, Cripple is behind, and PDM is behind. So he was arguing maybe this is this is not a temporal series. In this case, it's a spatial series. It might just something in common, which is striking. I don't really understand that, but it's very interesting. So,
but you agree that conceptually, if we're talking about a sequence of cell types being born from a single place, there's no other ways to make that than have somewhere somehow something that's temporally expressed in a serial way.
Yeah, but it could, as you say, it could be purely provided from the outside, a clock that ticks in the development of the animal. So, but again, I want to emphasize also that, we have this beautiful temporal series. We follow on Chris Doe's temporal series, and those have nothing to do in common. It means they're completely different genes. They're just transcription factor. They're not the same at all. So which means in the same animal, two different organs have invented two very different temporal series. So it's maybe a way to do that. And, there are not very many transcription factors, and they're all interconnected in some genetic network. And basically, nature can take advantage of these networks and just recruit them for doing something very essential, which all knows that making neurons the most essential thing in life. So.
Okay, a question from Urvi Mishra. What role would epigenetic factors play in encoding the temporal specification of different cell types?
Okay, so epigenetics just I don't know what epigenetics mean. I mean, you mean chromatin marks, and chromatin marks come from the transcription factors that bind to DNA and change the chromatin structure or the marks on the histone. Now, there is actually a beautiful work that came out from Chris Doe's lab, where they use this technique developed by Andrea Brandt, which consists of basically doing some epigenetic marks on developing, and they try to figure out what I just described to you, the different neuroblast at different location. They go to the 10 temporal series, but they produce different types of neuron. And the problem is that the genes that mark those neuroblasts early on, like in our case, don't remain expressed in the neuron. That means they are just expressed, not even in neuroblast, expressed in the neuroepithelium that give rise to the neuroblast. And yet, those change the output of the temporal series. And they show beautifully well, actually, that these special factors that are labeling the neuroblast actually changing the chromatin in a permanent way. In such a way, now some genes are open for business, some are not open for business, and the same temporal series gene will only turn on some gene in one case, not in the other. So you have a memory of where you were, even though you no longer know where you are, because the genes that specify where you are no longer expressed, but they remember where they were expressed. It's a paper in Life from Chris Doe's lab. It's a very nice piece of work.
Okay. Maybe one last question by Sonia Sen. Do you think Notch rewrites?
Is the one who did this work. So.
Okay, by Sonia then. So very specific question there. Do you think Notch rewrites chromatin landscapes to ignore spatial information?
Yeah, so it's a good suggestion. And, Notch is exactly like those spatial factor in the sense that, Notch is something which is expressed very transiently. At the time when the GMC divides, he produced two neurons. They just have lateral inhibition. One expresses Notch, the other one doesn't express Notch. And then it goes away. And then this is a long-lasting effect of Notch on the fate of the neuron that comes out from that. So So it's likely that indeed it affects, and again, because it's transient, it must affect the chromatin structure to be able to keep some memory. And so one idea will be that indeed the fact that Notch is there might be make sure that the transcription factor ignore or take care of the special information coming out from that.
Okay. All right. I'm writing to Sonia so that she can drop maybe her paper there for those who are interested.
Okay. Yeah.
Okay. All right. I think we're reaching the end of our allotted time. Thanks for the nice discussion.
To coin the term gros pâté. Is it you?
Yeah. So no, it wasn't me. It was Esther Klinger. So
foie gras is forbidden now in New York, so not yet.
Yeah, but gros pâté is more like on the beach, right? You put your stuff in.
Okay.
Anyway, you can use it any way you want. All right, Claude, thank you so much. It was
to all of you. Thank you for listening, and I'm glad that so many people showed up. Actually, so
yeah, it was great. So we'll see each other. I don't think I'm the one presenting next week. If I'm not mistaken, it's going to be Alain Chedetal, and the guest will be Sten Linnarsson from the Karolinska Institute. Until then, I wish everyone a good week. Stay safe, take care, and see you around. Bye-bye. Bye.
Keep distance and get vaccinated.